All right, good morning, and I hope you're able to see the PowerPoint that I have shared, and I'm clearly audible to all.
Yes, ma'am.
Okay, thank you. So, Piyush, welcome back to the class. You've been absent for quite a while. Hope everything is all right at your end.
Yes, ma'am. Yes.
Okay, so
Today I
as
was working in the lab and so it's very difficult to manage coursework and the work.
I am. Yeah, I agree. I agree. All right. So, I will begin by apologizing for a wrong statement that I made last in my last class. So, I have explained that the polymerases always work by extending the DNA strand or the RNA strand from the 3' end onwards. So 5' to 3' is the direction for synthesis. And then I was trying to think if there is any exceptions, and I do not know why I ended up saying that in tRNA, when the terminal CCA is added, maybe that's when it happens, but even there it does not. That's again a 3' extension. So, as far as I know, there is no polymerases that can work in the reverse direction. If anybody finds out an exception, please do let me know. And with that, let me move on to today's class. All right. So we have seen that there can be multiple polymerases, and DNA Pol 1 was the first polymerase to have been identified. And DNA polymerase 1 is also very simple polymerase. So let's go back to try and understand DNA polymerase 1 once again. So DNA polymerase 1 is one long single polypeptide chain, but it has multiple functionalities, so multiple motor activities in this single polypeptide chain. So if you look into the literature, they would explain to you the DNA polymerase 1 catalytic structure as consisting of a palm, fingers, and a thumb. So the idea is that the palm is the region that is responsible for the catalytic activity of the polymerase. So polymerization is going to take place from here. This is going to help to guide the DNTPs towards the catalytic center, whereas the thumb is going to be the region from where the nascent single-stranded DNA is going to be released. And in this entire domain of DNA polymerase 1 is also present the exonuclease activity, that is the 3' to 5' nuclease activity of DNA polymerase 1. But DNA polymerase 1 is also known to have a 5' to 3' nuclease activity, and that resides in a separate domain. So if you look at the structure of DNA polymerase 1, you see that here there is a separate domain, and this domain is linked to the main DNA polymerase 1 by 16 amino acids, which do not really take up a known secondary structure. So this part, when it is cleaved away from DNA polymerase 1, the rest of the DNA polymerase 1 is called the clinofragment, and this happens when you treat the DNA polymerase 1 with the protease subtilisin. And the clinofragment therefore will have the polymerization function, will have the 3' to 5' proofreading function, but will lack the 5' to 3' nuclease function because that resides in the separate domain. So with this introduction, we will go into today's lecture. So what I'm trying to do in today's lecture is give you an idea of how you can study these functions in real time and on a single molecule biophysics level. So the idea is that this one single subunit, this one single protein of DNA polymerase is going to be able to sit on the DNA and polymerize it when required. But this has a specific nuclease activity. And the nuclease activity is important for it to do some kind of repair functions when it sees nicks, or it has to get rid of the primers which are present at the lagging strand. So there are primers, then there are those primers are used for synthesis of discrete DNA strands in the lagging strand. So those then have to be replaced, the primers have to be replaced, and the rest of the DNA needs to be extended, and then there will be ligase that would attach or ligate the two ends of the DNA. So this is the normal process, and that for this to take place, the DNA polymerase 1 needs to identify the nascent DNA that has been produced, elongate it. In that fashion, it goes on, and it could get rid of the primer. It starts to elongate through that until it reaches the other DNA, the other nascent DNA that has been produced. It stops, and then the ligase will come and ligate the two strands. So for this function, it needs to work as a polymerase. It also needs to work as a nuclease. Suppose it finds that there is an error somewhere, it needs to get rid of it, or it is moving through, and it can also chop off these nucleotides from this end and just keep polymerizing the DNA. So there is a fine balance between its polymerization function and its nuclease function. The question is, how does the polymerase do both these functions simultaneously? We could think of multiple instances. For example, it could be that there is a polymerase that is polymerizing DNA, and then it finds that there is a place where it needs to act as a nuclease, and it can switch to the nuclease function. Switching to the nuclease function would require some sort of a conformational change, because it needs to move from the catalytic domain here, which is now bound to the DNA, to maybe the domain which has the nuclease function. And so some sort of a rotation, some sort of movement of the DNA polymerase must take place. And this could happen again by not leaving the DNA strand. It's still attached to the DNA strand, or it leaves the DNA strand and attaches back, but this time it attaches with the nuclease domain. Or it could be that this polymerase does not really go into this function, but rather a second polymerase which now has the nuclease binding to the DNA can come and attach. So there are multiple in scenarios that we can imagine, that more than one single molecules of DNA polymerase might be doing this function, that it polymerizes. Then when it has to act as a nuclease, it switches to the nuclease domain that now is bound close to the DNA. Or it could be that this falls off because it cannot polymerize anymore. It falls off, and a new polymerase attaches there, which now has its nuclease domain in the correct conformation or in the correct position, so that it can act as a nuclease and remove the bases from the 5' direction. So it can act as a 5' to 3' nuclease. So multiple scenarios can be thought of. And how do you distinguish which of these scenarios is correct? So this is one single paper that I'm going to discuss here, and the methodology that they have used is already known to us. It's FRET, it's Foster Resonance Energy Transfer. And I've already discussed Foster Resonance Energy Transfer, but I would like to emphasize two things once more. So for Foster Resonance Energy Transfer, the electronic configurations of the two molecules, the two fluorophores, the donor and the acceptor, has to be matched in such a way that the emission spectra of the donor must have an overlap with the excitation spectra of the acceptor. So the more the overlap, the more would be the energy transfer. The second criteria is that the two fluorophores, the donor and the acceptor, must be in close proximity. The closer the proximity, the more would be this energy transfer. And so the energy transfer falls in the order of distance to the power 6. So it's a very sharp fall if the two molecules go far apart. So even a little change in these distances would give a dramatic change in the fret intensities. So these two properties makes fret a very useful technique to study molecular distances. How close are two fluorophores, or how close are two molecules, or how close are two events. Even that can be parsed because of the property of this, the fret uh, or the energy transfer having an inverse relationship with the distance between the two fluorophores and inverse distance to the power 6. So in this particular experiment, what the authors did is they devised, or they were looking at a number of instances where DNA polymerase 1 can actually function. So you have, so this is a classical case where there is a primer, a short primer, and downstream of the DNA there is a double-stranded DNA, but in between this particular region is not double-stranded, it's single-stranded, but there is a primer. So instead of primer, you can also think of it as being another little double-stranded piece of DNA here, and in between there is a single-stranded stretch. And so DNA polymerase 1 can elongate this, can polymerize the DNA along this. As it does so, it could lead to a so this base pair might dislodge just because of thermal fluctuations, just a little bit. It could dislodge just a little bit. And so the DNA polymerase 1 might end up extending even further, and you could have this sort of a flap-like structure of single-stranded DNA, which does not polymerize with this template strand anymore. So just a little bit of thermal fluctuations could give rise to a structure like this. Alternatively, you could also imagine a structure like this, where the DNA polymerase, after it has created this flap, maybe it's dislodged from the structure, and now you could have a thermal fluctuation here, just a tiny little bit of thermal fluctuation, so that this strand forms a base pair again with the template strand, whereas now you will have two flaps, just one base pair or two base pairs of differences between these two strands, giving you a double flap kind of a structure. You can also imagine another scenario where the DNA polymerase 1 elongates and then perfectly stops at this point where it realizes that it can't go any forward, there is already a double-stranded DNA, and it falls off. And that's when the ligase would come and seal it. But what happens to these structures? If there are structures like these, then the DNA polymerase 1 must act as a 5' to 3' nuclease, and it needs to chop off these nucleases in order to create a structure like this for the DNA ligase to come and bind and create the double-stranded piece of DNA. So the idea is that when the DNA polymerase encounters such structures and it needs to switch from the polymerase mode to the nuclease mode, to the 5' to 3' nuclease mode, how does it do it? Is it a spontaneous switch, or is it that you need another molecule that comes and does it? How efficiently is DNA polymerase able to do these switches? So that's what they were interested in. And so the authors decided to make these DNA structures in vitro. So they created single-stranded DNA pieces and then annealed them to create these different structures. So these are the scenarios that we have talked about before, and so they created each of these scenarios, like a single flap. This is the single flap. Here you have a double flap. A double flap arises because you can have just a little bit of thermal fluctuation, so that one base pair of this strand that was being synthesized does not form, rather the previous strand anneals again, and so you can have a little bit of mismatch on both the strands. So you can have a double flap structure, and you can understand that these two structures can also sort of interchange between themselves because of thermal fluctuations. You could have one structure moving towards the second, and that's possible. So you could have a mixed structure kind of configuration. You could have a clear gap. So you have one or two nucleotides that have not base paired because there is no corresponding nucleotide that has been added. So there is a clear gap. Or you could have a situation where you've added a single-stranded DNA and another short piece of single-stranded DNA that anneals with this DNA, so giving you a primer kind of a structure. So these are the various structures that they designed and produced in vitro in the laboratory, and now they use this and attach them to a glass slide. Now, we have seen this repeatedly whenever they are trying to do single molecular biophysical studies. You need to be able to attach pieces of DNA or even actin or tubulins to glass slides so that you're able to monitor fluorescence. Because you cannot do that, you cannot do single molecule studies when they are floating all around. You'd rather have them attached on a glass slide so that you're looking at events at one particular position on the glass slide. So they added this thymine 10 DT residues, and then that was attached to the biotin. So using biotin streptavidin chemistry, they were able to attach these pieces of DNA specifically, whichever they chose, on the glass slide. Now, the next idea that they used was the idea of using two different fluorophores. So one fluorophore, suppose this is the structure they start with. I'm saying suppose, it could be any of these structures that they have chosen. Suppose this is the structure that they have started with. And the idea is they would like to look at it as the polymerase goes around polymerizing this piece of DNA. So they attached an acceptor fluorophore molecule to the DNA polymerase, very close to the region that is responsible for the polymerase activity. So somewhere near the palm, that is where they attach a acceptor fluorophore. And the donor fluorophore was attached definitely to the double-stranded DNA structure, from where you find that it, after a while, there would be a single-stranded stretch where it can go and polymerize the DNA. So this was kind of the configuration that they used. Now, these are some nitty-gritty details that I've added here that was present in the paper. So what are all of these? These are actually names of the fluorophores that they have used. This Alexa 488 simply means that it is excited by a laser line that has a wavelength of 488 nanometers. So nothing much to worry about here. These are just names of fluorophores. This is a donor fluorophore and this is an acceptor fluorophore, and they are used because they have considerable overlap of the emission spectra of the donor versus the excitation spectra of the acceptor. So another thing to remember here is that in the DNA polymerase, when they have added the fluorophore, they have specifically added it to the clinofragment, not to the exonuclease fragment, because they would like to observe the polymerase activity. So the acceptor fluorophore is very close to the palm region, the region that is known to act as the catalytic site for the polymerization reaction. Okay, so here again you find another structure where they are explaining the same concept. So you have one base pair, one single base pair in the double-stranded DNA structure, which now has a donor fluorophore, and the acceptor fluorophore is on the DNA polymerase 1 clinofragment, very close to the catalytic site. So the idea is that if the DNA polymerase 1 is going to act as a polymerase on the structure, then these two fluorophores must come in close vicinity. And as the polymerase functions, the distance between these two polymerases would increase. So when they did this kind of an experiment, they observed these donor acceptor fluorescence what should I say, spectra over time. So this is over time, and the red here indicates the fluorescence from the acceptor. The green indicates the fluorescence from the donor. So as soon as they start this experiment, when they load the DNA polymerase onto this slide, they find that initially the fluorescence of the donor is very high, the fluorescence of the acceptor is very low, because the acceptor, you're not specifically exciting the acceptor would emit its fluorescence once it is in close proximity of the donor. Is everybody with me?
Yes.
Yes, okay. So initially when you have loaded, so you have the slide, you have all the DNA strands, and now you're adding the polymerase. So it would take time for the polymerase to come and bind to this DNA to find the region where it needs to start polymerizing. So when you add it at that instant, you do not have an acceptor fluorescence. But when the polymerase comes in close proximity to this donor fluorophore, that's when you see the acceptor fluorescence rise, and the donor fluorescence would actually go down because the donor, because of the phenomena of FRET, so because of energy transfer, you no longer see the fluorescence of the donor, but you start to see the fluorescence of the acceptor. Now, if you make a ratio of the intensity of the donor, acceptor fluorescence versus donor fluorescence, you get something called this FRET line. So what this indicates is, with time, when does the fluorescence resonance energy transfer actually take place, and when does it go down? So they were able to see such signatures. So this is one signature for one experiment. This is another signature. So from all of these signatures, the one characteristic thing that you can observe is the following. You see that there is a peak for the FRET intensity. So somewhere here there is one peak, somewhere here there is another peak, here there is another peak. So every time you have a very high acceptor fluorescence, you see a corresponding peak for this FRET ratio. But there are two distinct peaks that you can observe. One peak shows you a FRET efficiency of nearly 0.7, 0.8. So if you see here the ratio, because you're doing a ratiometry here, what is the ratio? Ratio between the fluorescence of the acceptor versus the fluorescence of the donor. So that's giving you a FRET ratio. Now the FRET ratio, the maximum ratio that you can have is 1, and the minimum is 0. So when you do not have, when you don't add any of these polymerases, you have a FRET efficiency of 0. But when you add the polymerase, and when the two fluorophores are really close, the maximum FRET efficiency that you're able to get is 0.7 to 0.8. But you also see another peak here that comes to about, say, 0.6. So what does this indicate? And you see that again here. You see that sometimes you have an efficiency of 0.6. Sometimes you also have an efficiency of 0.8. This indicates that there could be two states or two probable configurations in which the DNA polymerase is now bound to the DNA. In one configuration, the distance between the donor and the acceptor is in such a regime that you have a higher FRET efficiency compared to the other. And that may be possible. So this is again a hypothesis that may be possible if the DNA polymerase is bound in this orientation, where the polymerase is really in close proximity with the donor. And the second configuration that may be possible is when this nuclease domain is in close proximity to the donor. So if this is in close proximity with the donor, then definitely the distance between these fluorophores would be a little greater than this case, and therefore you could have two peaks. So in both the instances, this molecule is going to be close to the donor fluorophore, but the orientation of the polymerase might differ. So this is a hypothesis. How do you prove this hypothesis, or how did they go about proving this hypothesis? That indeed the two peaks that they observe here, the two distinct FRET efficiencies that they observe, one is about 0.8, one is about 0.6, is indeed because the DNA polymerase now exists in two different ways on the DNA. One in which the polymerase palm is wrapped around the DNA in such a manner that its acceptor fluorophore is close to the donor fluorophore. In the other case, you have this additional domain which is now close to the DNA, and therefore this palm domain is a little further away. That gives you the second peak. So one easy method to do this would be to chop off this, right? Because we know there is something called the clinofragment, where you can add a protease and remove this 5' nuclease domain completely. That would give you the polymerase with the palm domain, but without the ability to have the second subdomain that can interact with DNA. So if that is the case, if the two states, one of 0.8% efficiency of FRET and 0.6% efficiency of FRET, if these two states are because the polymerase itself is in two different conformations on the DNA, one in which the 5' nuclease subdomain is actually interacting with DNA. If you remove that, then there is no way this domain would interact. So if one of these efficiencies was related to this domain, then you would no longer see that efficiency state. Is that okay?
Yes, ma'am.
Okay. If that is true, then here is the case that they found. So when they used the polymerase 1 without the associated subdomain of the 5' to 3' nuclease activity, so which is just the clinofragment, they find that the maximum FRET efficiency they observed was always 0.6, which indicated that the 0.8 efficiency what they were observing was because of the DNA polymerase part, was because of the exonuclease part that was coming in close proximity with the DNA, and therefore somehow this catalytic domain, the fluorophore that was close to the catalytic domain, was in close proximity with the donor. So from this experiment, this little part here, you can, and by looking at this, you can clearly understand that indeed there can be two states in which the DNA polymerase can bind to the DNA. And the DNA polymerase can actually switch between these two bindings. Why? How can you understand that it is a switch and not a binding and then a fall off? You can understand that by looking at this little curve here. So if it had been a binding and a fall off, you would have seen a case like this. So you would see an increased FRET and then a drop because it fell off, because the DNA polymerase just dislodged from DNA. So you would have seen the FRET ratio come down to 0. But you don't see that. Instead, you see that there is a situation where you find there is a FRET efficiency of 0.8 and then it drops to 0.6, which means the DNA polymerase is able to bind to DNA and then switch between the polymerase subdomain and the 5' to 3' exonuclease subdomain on the DNA. So it can switch conformations on the DNA while it is still bound. It need not fall off. So this also increases the process. This also indicates the processability of the enzyme, that it can do both these functions while it is still bound to DNA. It need not dislodge. So far, is it okay?
Ma'am, I have a question here.
Yes. Yes.
So, ma'am, as you said that fret efficiency decreases as the distance increases, right? So polymerase will be moving onto the primer, sorry, onto the single strand that is far from the green fluorophore. So isn't that affect the intensity of fret ratio?
Yes, that will. That will. You're absolutely right that as the polymerase keeps moving on the DNA and as it keeps, because there is just one point of the donor fluorophore, just they've designed it in such a way, and this can be done. You can design it in such a way that you just have one point of adding a donor fluorophore on the entire DNA stretch. And when the polymerase loads on the DNA, and is in close proximity to this fluorophore, you see fret. But as it moves away, you would see a drop in fret intensity. And you're absolutely right. You're absolutely right with that. But you have to understand that the problem here, or the situation here, is that you do not have very long stretches of DNA. So I think this was about 20, 30 nucleotides that they have designed. So not very
So
long
10
stretches.
nucleotides as we have seen,
10
10
nucleotides
pp.
is the nucleotide that they've used to attach this particular
Oh,
stretch
okay,
to the glass slide. So that
okay,
DT10 is actually at the end, and that has not been shown in the figure. That's an extra information that I gave just to show you that there was some way to attach this to the glass slide. But the double-stranded DNA with the flaps or the gaps or whatever they've designed are short stretches. So you see the DNA polymerase binding, moving, and then what happens? Once it's at the end, it might even fall off, or once it's done its work, it falls off. But then there would be another DNA polymerase that would bind. So because this is in solution phase, you're looking at it over time. So let me go here. You're looking at it over time. So you are not observing one single molecule, you're observing one single molecule event on one single strand of DNA, but you're observing over time, which means that on that DNA strand, you will have repeated binding, loading, falling off. So you could be observing that too over time. So this signature that you see here is a binding coming together of these two fluorophores and then a sharp drop, and perhaps the DNA polymerase has fallen off. You see a binding here and then a sharp drop, which might again indicate actually the DNA polymerase has fallen off. Whereas here you have a binding and then you have a second state that the DNA polymerase has taken, presumably because of this formation of a single flap here, because it needs to now move from the polymerase function to exonuclease function. And then what happens next once it has done this? It should fall off because it's not a very long stretch of DNA. So you could have another event of another polymerase that can bind here and do some activity.
okay, ma'am.
Okay, so the idea is this: you are able to, under a microscope, you're able to look at fluorescence on this particular strand of DNA because you are able to specifically locate the DNA strands at distinct distances on the glass slide. You can do that. But you cannot control the number of polymerases that would come and attach to the DNA. Over time, you could have multiple such attachments, detachments, and you're going to follow each of those events. Of course, each of those events is a single molecule event, but you'll have multiple such events. And given the short length of this DNA, you are expecting to see multiple events. The more events you record, the more you would understand what is exactly happening at this point.
Okay, ma'am. Thank you.
Thank you. Okay. So, well, let me go here once more. So here they have used another mutant. So one is clinofragment. Clinofragment itself gives you a beautiful indication that these two distinct states of fret efficiency that they were able to observe, one of zero point 0.8, one of zero point 0.6, is because of the presence of two distinct functions of this polymerase. So this polymerase is able to produce two distinct motor activities. One is polymerizing motor, and one is kind of a depolymerizing motor. So this exonuclease activity, these nuclease activities, they are all depolymerizing. So something which is double-stranded needs to be chopped off to create a single-stranded stretch. And both of these activities are present in this single polypeptide chain. And both of these activities could go hand in hand in the same molecule. So the molecule need not dislodge and do it. Once it is acting as a polymerase, it could switch its conformation on the DNA in such a way that now the nuclease activity starts to become prominent on the DNA. So this was clearly seen in their experiment. And which of these efficiencies is due to the 5' to 3' nuclease activity? So you take just the cleaner fragment and you're able to see that one of the FRET efficiencies, 0.8, is absent. So that could be because of the 5' to 3' nuclease activity. Now here is another interesting sort of mutation that they used, and this mutation is known to make the DNA polymerase 1 deficient in the 3' to 5' exonuclease activity. So not the 5' to 3' nuclease activity which you delete and you get the cleaner fragment, but the 3' to 5' nuclease activity. So that can be so what is that? So that is the proofreading activity. So as it keeps adding nucleotides, it checks if it has added the correct nucleotide. If not, it will remove one. So that is the proofreading activity, and that would be absent if the polymerase has this kind of a function. So this function is therefore something that would slow up the polymerase activity, is it not? Because every time if it has to add some nucleotides, then go back and check, then add some nucleotides and go back and check, then it is something that sort of slows down this polymerase activity. And so they also used this particular mutant, and what they found is when they use this mutant, this species of 0.6 efficiency of FRET is actually no longer present, which means that this mutant somehow this proofreading, if you remove this 5' to 3' proofreading activity, it makes the polymerase sort of super fast, and it goes on adding all the nucleotides and acts more like a polymerase, but it also inhibits the sort of it kind of enhances its activity of the 5' to 3' nuclease. So it no longer has the activity of the proofreading function, but the dominant function of the 5' to 3' nuclease activity can be seen here. So no longer just goes back, reads it, but if it encounters a structure like this, then it automatically switches to the conformation where the nuclease activity predominates, and therefore you always see this 0.8% FRET efficiency. So this is something very interesting that came out in this study, that if you leave out this proofreading function completely, what is proofreading function? So it keeps adding nucleotides and it goes back and checks. If it cannot go back and check, then it would be a very fast polymerase. But if it encounters structures like this, the single flap, or it encounters structures like this, the double flap, then it always essentially acts as this nuclease, removing the flaps from the 5' end. So 5' to 3' exonuclease activity then dominates, and you see always a signature with a FRET efficiency of about 0.8. So this is just an additional information that they obtained using some of the mutants that they were studying. Okay. So this is one way of representing the data that they have obtained. Essentially what it shows is the two states that they have hypothesized. One state is where the DNA polymerase is acting as a polymerase. One state is where it is acting as a 5' nuclease. So if there are these two states, then in a wild-type polymerase, you always find that you observe both the states. You observe the states of polymerization, you observe the state where it is acting as a nuclease. But if you give it a double flap, what is a double flap? This is a double flap. Sorry. this is a double flap, and this is a single flap or a downstream flap. So if you give a wild type polymerase a double flap, then the nuclease activity is something that you see predominantly. Whereas if you just have a downstream flap, then the polymerase activity predominates. Intuitive. So if the DNA polymerase were to load onto this kind of a structure, it definitely cannot polymerize until it finds a good base pairing at the 3' end. Until it finds that, the first thing that it would do is to chop off this piece of DNA using its 5' to 3' nuclease activity, perhaps get a little bit of this one nucleotide that did not base pair to base pair with this strand, and then act as a polymerase. So that's exactly what they see here. So if they have this double flap, the polymerase activity is very little, but the exonuclease activity predominates. Oh, sorry, we have a structure here. But this is, I have reversed it. So double flap is this, downstream flap is this. But if they have a structure like this, it's possible that it can keep elongating, keep dislodging the strand, and then act as a nuclease. And that's exactly what you see here. If you add a clinofragment, the clinofragment would not have this nuclease activity. So you do not see the structure coming up ever. And here, you do not see that FRET efficiency coming up ever. For this mutant, the situation is mostly like the wild type when it comes to the downstream flap, which is this, because it keeps adding the nucleotides, and works as a polymerase. But if it encounters something like this, now this does not have a 3' to 5' nuclease activity, and you see that you're able to see both the nuclease and the polymerase activity almost at the same efficiency for a double flap. But a downstream flap, if it is this, then it acts mostly as a polymerase. Okay, we now go to a different kind of a setting. So far, the fluorophore was attached to a polymerase subdomain. What if you add the fluorophore to the nuclease subdomain? Then you should be able to see some other kind of FRET or something else, some other situation. So let's see what happens if you add the fluorophore. Instead of adding it to the polymerase domain, you add it to the nuclease domain. And now you try to see what's actually happening. And because you're adding it to the nuclease domain, there is no point in adding the donor fluorophore here, because the nuclease domain would be acting where there is a kink, or there is a mismatch, or there is, in this case, there is a flap-like structure. So they add the fluorophore very specifically to these structures, and they observe that when the polymerase is not loaded, you again see that there is no FRET. But once the polymerase is loaded, there is a FRET at a very high efficiency. So you see high efficiency of FRET every time it is loaded. That's the first observation. Just like previously, if you have not added the polymerase, there is no FRET. The moment you add it, it switches on instantaneously. And it switches on, and again you see these two states. Okay, so this is the same thing I have explained. So again, they find the two states, and the two states of these FRET efficiency that they observe is again linked to the two ways in which the polymerase was moving. So the idea here is this: if you have seen using a donor fluorophore pair, and by specifically labeling the catalytic domain of DNA polymerase 1, you have been able to observe the two FRET states, then the question arises that is the labeling the very event that you've labeled the catalytic domain of DNA polymerase 1 is that itself creating these two Fred states. It may so happen that your labeling function, that the way that you have labeled it, has artificially created these two states. The two states actually do not exist, but because you've labeled it, you've created some local conformational changes that makes the protein molecule lock into some sort of a second state, even though the first state is more predominant. So how do you counter the argument that the very fact that I've labeled a molecule has artificially created these two states? You can counter this argument by moving the label from that point to some other point and to show that you're exactly getting the same two states. These two states are not quite different but exactly similar. So that's what we've done here. The reason for doing this is two-pronged. One is to counter the argument that you have artificially created the two states. The two states do not exist. You've artificially created it because what you're observing is just two Fret efficiency states. You are arguing that the two Fret efficiency states are because the two subdomains of DNA polymerase 1 are able to bind and interact with DNA, and therefore this different binding is creating different distances between the donor receptor fluorophores, and you're able to see the two states. What if this is not the case, but then the polymerase is locked in one state, but because you've attached the fluorophore, there is a probability of creating another state just because of adding the fluorophore. And that you're observing changes between those two states because you've artificially added the fluorophore. So what they did was they simply switched the fluorophore, the acceptor fluorophore, from the polymerase subdomain to the 5' nuclease subdomain. And again, they see the same kind of two different states appearing, which goes to counter the argument that the two states arose because of specific labeling. Because if it was just because of the labeling, you wouldn't see the same Fret intensities of 0.8 and 0.6 coming up. The intensities would have been different because now the conformational changes would have been different if it was indeed induced by the labeling. So this is kind of, you can see it's one of the controls in their experimental design to ensure that their interpretations are correct, that they are not introducing an artifact in their experiment. So again, using this, they went on to check their previous results, and again they find very similar things, that if you have say a double flap, then you would always see the nuclease activity predominating over the polymerase activity, which has to be the case, because how would the polymerase act here, because it doesn't have a 3' end which is tightly bound to the DNA. Whereas if you have a case where you have a single flap, then the polymerase can work efficiently because it has a free 3' end and it can elongate before it tries to dislodge this with a 5' nuclease activity. So they were able to reproduce the same data again using this kind of a labeling strategy, where they simply shifted the label from the polymerase subdomain to the 5' nuclease subdomain. Okay, so this is the paper that I have presented to you, and you can have a look at this paper. It's a very recent paper, but it uses a very simple technique, something we have seen that before, and it uses it to clearly illustrate to you that the DNA polymerase 1, the two functions of DNA polymerase 1 can go on without the DNA polymerase having to dislodge itself. And that is clearly seen, again I'd like to point out, clearly seen in this spread, because this is a case of either the DNA polymerase 1 literally falling off or going really far off. Going far off is not a possibility in this case because this piece of DNA is literally short. So rather what you're observing here is the falling off of DNA polymerase once it has done its function. But here you see the DNA polymerase 1, it does not fall off, it rather switches to the second state. So switches to the second conformation. So from polymerase to nuclease or nuclease to polymerase. These kind of switches are possible. You see that again here. All right, so I've come to the end of today's lecture. This was just a kind of I just tried to highlight to you how the functions of DNA polymerase can be observed under a microscope. And tomorrow we will not look at polymerase, we'll look at something completely entirely different. And tomorrow would be literally the last class before Friday. Friday, I would try to do something different, completely different, not quite taking a class. We'll try to do some activity on Friday. But tomorrow would be the last class where we are, where we'll look at engineered not polymerases, but engineered motors. Some sort of futuristic design again. We'll try to see how those can be used to walk on DNA, how things can be used to look at local pH changes, etc. So if you have any questions on what we have seen so far, I'll be here for another 5-6 minutes to answer. Oh, I think I have more, about 10 minutes.
Ma'am,
Yes, sir.
the second red signature we are observing in the above figure, this is for a longer period of time. So, is this due to multiple attachment of polymerases?
This
This is for a longer period of time. The second red signature we are observing in the above figure.
so you see here you have an event where you can assume that as long as this signature is there, it is attached to the DNA. But when it falls off, you do not see the signature, so you can assume that the DNA and the polymerase are not bound. Why do we assume this? Because we know that when you do not add the polymerase, this is the signature that you will see.
Ma'am, I'm referring to the above figure,
This
figure
figure?
B.
Yeah,
Yes, ma'am.
this. Okay.
Here we have two red signatures.
Yeah, we have multiple red signatures actually highlighted too. So you have here, you have here, and you also have a short one here that did not seem to materialize, and maybe the DNA polymerase just fell off.
So the first one is for a shorter period of time, and the second one is for a longer duration of time. Is this due to the multiple attachment of polymerases happening on a single double-stranded DNA molecule?
It is possible. It is possible because, as I said, you can have all possible events, all possible stoichiometry is happening on this class slide. Why? Because you cannot control the number of polymerases that would load on the DNA. It's also possible that because of the presence of such a structure, the DNA polymerase may have stalled. So stalling is something that you've seen previously when you have looked at kinesins or such motor proteins when they were walking on tubulin. So if you remember, did I show that? I think I did. So if there is a junction of tubulins or even actin and the myosin or kinesins which are walking encounter a junction of these tubulin or actin tracks, and then at one point when they encounter these junctions, they stall. They try to sense if they should keep moving on the same strand or they can flip and move on the second strand. And sometimes they walk on this strand, sometimes they flip on the second strand. So this is called stalling. They kind of stop at that point. So sometimes the polymerase could also stall at this region, but definitely that cannot be this long. So it is most probably perhaps a combination of stalling as well as a combination of another molecule that has come in the vicinity. Could be both the events. Very difficult to say which of this from this signature alone.
Ma'am, I have a request to make.
Yes, Farman.
Ma'am, if we could please talk after the class for a minute or so.
If we could.
If you could have a discussion of a minute or so after a class, like if you have a time to discuss something.
Yeah, I'm all the time. I have all the
Okay.
time. It's just I don't know if you all have the time. I have all the time. I'm
Okay,
here to
ma'am.
discuss. No issues. You can discuss online, offline, whenever you
Ma'am,
want.
just after the class. Just after
Yes,
the
yes,
class.
yes. I'm here. I'm here.
Okay, ma'am.
Anything else? No, no other questions. Okay, if there are no other
Ma'am,
questions,
I have just one concern. Then as
you
you said that futuristic models that we have studied will not be included in our syllabus of my major, right?
no, no.
Okay.
No, no, no. But futuristic design will not be included in your exam because I can't really ask you something that we really do not know. Futuristic
Yes.
design, as the name suggests, is some things that are still
Hypothesis.
being explored. So there
Okay.
cannot be questions on that. And you've already done a little assignment on it. So no.
Sure, ma'am.
So do not worry about your exams. It's going to be open book, open source, and you've seen the pattern of the minor exams. So it's going to be very similar. You'll be asked to design an experiment or to analyze the data. So it's going to be simple. If you have attended your classes, you should be fine.
Okay, ma'am.
Okay, so if there are no more questions, I think we can stop for today. Thank you, students, and we will meet again tomorrow.
Thank you.
Thank you.
Thank you, ma'am.
Thank you. I'll stop sharing.
Ma'am, shall we talk now?
Yeah, yeah. Just let me stop recording. Or do you want to record this?
No, 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 ma'am.